All right, so for anyone who doesn't know me, my name is Lauren Moore, and I'm the Chief Scientific Officer at the National Ataxia Foundation. Just a couple housekeeping items before we get started. If you can please keep your microphone on mute throughout the presentation, just to limit some background noise. And also throughout the presentation, if you have any questions at all, please feel free to type those into chat, and we'll try to get through as many of the live audience questions as we can at the end of the presentation. I also need to quickly announce that there has been a last-minute change in the speaker today. Unfortunately, Dr. Biondi will be unable to present today, but I'm excited to welcome two of his colleagues from Seelos Therapeutics, Doctors Claudia Moore and Raj Mehra. It really is an exciting time for the ataxia community. For the first time ever, there are multiple drugs that are actually under consideration or will soon be under consideration by the FDA for the treatment of ataxia. And there are also multiple clinical trials launching this year for spinocerebellar ataxias and other ataxias. So I'm very, very excited today to welcome our partners from Seelos Therapeutics to talk about one of those trials that's starting this year. So without further ado, I'll turn it over to today's PrepRARE speakers. Thanks.
Thank you, Lauren. Thank you, everyone, for joining. My name is Dr. Raj Mehra. I'm the founder and chairman of Seelos Therapeutics. Joining me will be Claudia Moore as well. She's our head of our clinical operations for this study, and she will be controlling the slides. With that, Claudia, can we have our first slide, please?
Can I actually have the host give me the rights to present the slides? It's giving me the...
That'll be helpful.
Yeah.
So, our job will be to leave at least 20 minutes towards the end to have many questions that we can, because we want to keep it interactive, but also give you ample description of the study. So hopefully they'll answer most of your questions, but not all. And towards the end, what we will say, to the extent certain questions that cannot be addressed or you think of it later on, feel free to come back to us directly. We will splash the email that you can contact Seelos Therapeutics directly, or certainly our partner, National Ataxia Foundation, reach them, and through them, we will be able to address any questions that you may have later on. So with that, we'll get started. Thank you. So today we'll discuss the study called STRIDES. So it's a double-blind randomized placebo-controlled study to assess safety and efficacy of SLS005, which is an IV Trehalose, for the treatment of adults in Spinocerebellar Ataxia 3. This study, although we will discuss many pertinent parts, is supposed to be a global study. So we will discuss later, and Claudia will give you the details, that we are enrolling the studies not only in the US, but also in Latin America, Asia, Europe. And it is primarily driven by many patients in Asia community. They were willing to travel to the US to participate in this late stage study. And it behooved us to just open it up for the whole, the global community. So a little bit about Seelos Therapeutics. We are a clinical stage pharmaceutical company based actually in New York, Manhattan, right on Park Avenue across from Waldorf Astoria. And we are focused on CNS and rare disorders. And our other programs, for example, we are currently doing a pivotal study in suicidality in major depression. That's a registrational study ongoing as we speak. We also have a pivotal study going on for ALS, or also known as Lou Gehrig's disease. And plus we have some open label Alzheimer's studies going on in Australia and other neurodegenerative diseases. And plus we also have some preclinical programs in Parkinson's. So we are squarely focused on the CNS diseases. Next slide, please. Since we are a publicly traded company, we flag the disclaimers attached to any forward-looking statements that we may make today. So we do want you to consider disclaimers on this slide and on all our public filing, as well as on our websites. Next slide, please. So a little bit about IV Trehalose. So first of all, the IV Trehalose has not been approved for any indications worldwide, so it still is an investigational drug. I mean, we want to make that clear about that. And the other thing is that since the Trehalose is generally used in the food industry and it's widely available and considered generally regarded as safe by the FDA as well as the equivalent designation in Europe and elsewhere, it cannot be taken by mouth as a therapy. It can be taken for other things. By taking it by mouth is not equivalent to receiving Trehalose by IV infusion, and I'll get into it a little bit later in other slides. And also, so far, our experience has been that this has been very safe and well tolerated, and we'll give you the number of patients that has been exposed to this drug. Still, you may get some unexpected side effects that we may not be aware of, but we just wanted to flag it. Not everything is completely known, but there is enough known for us to indulge in this study. So with that, let's go into the details. So we are focused on spinocerebellar ataxia. You probably know more about it. It's a neurodegenerative diseases, but specifically there are 47 subtypes of SCA, ranging from SCA1 all the way into the 50s. And 35 causal genes have been identified that lead to this disorder. And primarily, what you have is the accumulation of abnormal proteins. But different abnormal proteins lead to different subtypes of SCA. And as a result, the FDA's position is that each subtype should be treated differently for clinical studies purposes. So our study currently is focused only on the type 3. This does not mean mechanistically that our drug does not work or will not work for other subtypes other than subtype 3. But currently the study is only for subtype 3, and the non-3 patients we will eventually do a future study. So stay tuned for that. But right now, based on the FDA's requirement, because they wanted for statistical purposes, analyze each subtype separately. So and based on the wider prevalence of subtype 3, we decided to do the subtype 3 first, and we will do the non-3 study in the future. Next slide, please. So a little bit about Trehalose. Our human bodies, it's all about amino acids and carbohydrates. So amino acids, there are 20 amino acids that define almost most of the naturally occurring proteins that exist in our body, and for carbohydrates, there are 6 and 7 carbohydrates that define most of the work. So Trehalose is actually a disaccharide where it's a very specific link between 2 glucose molecules. It's extensively found in plants and some animals, but it is not produced by humans at all. However, we do have an enzyme called trehalase that is specifically, its a specific function is to hydrolyze trehalose into 2 glucose molecules. And we humans have that enzyme trehalase, although the humans don't produce trehalose, the disaccharide. So that's just a developmental mystery, and I'll let the professors at Harvard and Columbia figure out why such as this thing happens. But because of that trehalase enzyme that primarily occurs in human gut, kidney, and liver, and there are traces elsewhere as well. So mammalian brains, for example, has been shown to have traces of trehalase as well. And as a result, any trehalose that's taken orally, it immediately gets hydrolyzed and is an ineffective therapy. And based on the prior data, we believe less than 0.5%, so 0.5% of ingested trehalose, will will survive. And even it's traveled all the way into the brain across the blood-brain barrier. It is a very ineffective therapy when taken orally. In order to bypass this trehalase enzyme, it must be given IV, and that's our therapy. So our therapy is given, through IV, and it's a weight-based dosing. Claudia will explain that in a second. And otherwise it's a very straightforward IV infusion, takes less than 60 minutes most of the time for infusion. And we have had patients that have been on it for 2 years plus, and it has been shown to be pretty well safe and well tolerated, but I'll give the details in the next slide, please. The key also for trehalose is if it remains intact, it can enter both into the muscles and the brain, and it's important that the therapy should be into the muscles, specifically for disorders like spinocerebellar ataxia. The way the mechanism works for trehalose is the mutation ATXN3, the mutation that causes the SCA3 creates a protein that is abnormal. And what that means is instead of the original protein being certain length, let's say X, it gets extra repeat of polyglutamine. So glutamine amino acids, there will be excessive repeats of 30, 40, 50, 60 extra repeats in the amino acids that's expressed by those mutated genes. And now that the protein expressed, the mutated protein, is longer in length, it cannot fold itself into a classical alpha helical native structure. And as a result, these mutant protein aggregates, they tend to aggregate, and those aggregates are cytotoxic within the neurons and cause movement disorders and all the effects that the patients see. There are a couple of ways how our drug mechanistically works. So number one, it stabilizes the proteins, even the mutant proteins, and inhibits the aggregation. Those aggregates are more cytotoxic, and you see that in the left bar. And second, there are two ways where these cells will recycle these mutant protein aggregates that are cytotoxic. One of the classical ways is the autophagy lysosomal pathways. And those pathways, the best way to understand that, they will literally recycle these mutant protein aggregates into amino acid components. And the way our drug works is it will enhance autophagy. And autophagy is the classic term for self-eating. And recently in 2016, when the Nobel Prize was given to a Japanese researcher that identified the genes responsible for autophagy, this area has become much more interesting for researchers, and all of us are focused on to see how we can avail ourselves to treat certain neurological diseases. And the way that autophagy basically works is by activation. It'll create a so-called phagophore around these abnormal proteins, which are also known as autophagosome, which will eventually degrade these proteins, and those the proteins will get recycled. And another way the pathway it works is our drug on the right side. You see the third bar. It will activate the transcriptional pathways, specifically the transcription factor EB, which will translocate from cytoplasm into the nucleus and activate both the lysosomal and autophagy genes to clear out these mutant protein aggregates. So that's a classic way to activate and clear out these mutant protein aggregates, and that's how our drug works. And remember also, there's a lot more work being done in these mutant protein aggregates that are causing these problems. They could be cytoplasmic, or they could be inside the nucleus. And to the extent it's inside the nucleus, there are another pathways called UPS, the ubiquitin proteasome system, which PrepRARE has also been shown to activate to clear those out to the extent these aggregates are in the nucleus. And that work was done elegantly by professors at Cambridge University in UK as well. And you can Google those things or come back to us and we can share those citations with you. In terms of what is the rationale, the mechanistic rationale for utilizing our drug in SCA? So activation of autophagy, reduction of abnormal protein accumulation that are toxic to the neuronal health, as well as the preservation of these cells. And more importantly, it is important that that the drug goes into the muscles, many axons in the neuromuscular junctions that ends in these muscles. You need to have the therapy into the muscles, which will lead to muscles difficulties that translates into walking and speaking difficulties. And that's why the smaller molecules like PrepRARE has a role to play. Now, all of us are also looking at the gene therapy programs differently. Currently, there are challenges, technological challenges involved with those gene therapies in the sense the capsids do not biodistribute well into the muscles. And as a result, we will eventually overcome those technological challenges, but that's the challenge right now it's facing. So even the gene therapy approaches currently, we believe they will be ameliorative in effect, not potentially curative, but we are working on it, okay? The next slide, please. The previous findings, let's talk about our drug. So it has been, the non-clinical exposures have been really shown, pretty benign. More importantly, prior to this year, we had 59 plus patients treated up to 24 months. And they were primarily in either a muscular dystrophy called oculopharyngeal muscular dystrophy, OPMD, disorder, as well as SCA3 patients that have used this drug before. But up until prior to this year, we have used half the dose, 27 grams IV per week in the clinical trials. And this year, we are also already have been doing a pivotal study for the same drug in ALS or Lou Gehrig's disease. That study started in March of this year. Almost 80 plus patients have been treated since then. And so far, it has shown it's relatively safe and well tolerated based on, I think, 3 or 4 safety committee meetings that have taken place and basically found there was nothing remarkable in terms of the safety and well toleration. So that bodes well. And what have we basically learned from these studies? That we have not found any cell toxicity, gene toxicity, organ toxicity. And simpler way to understand that is because the only known metabolites or breakdown of PrepRARE is glucose, and there's plenty of that in our body. So we have not seen so far, anything that's worth worth considering. So and also the patients could be on the other existing drugs. We have not identified any drug-drug interaction so far. Next slide, please. How does what happens when the PrepRARE is given? So first of all, it's weight-based dosing. So an average adult will get approximately 2, the IV bags administered within 60 minutes, within an hour. And but the short half-life of the drug is only 1.5 hours, which half-life just means within 1.5 hours, 50% of the drug is cleared from the blood circulation in that time frame. And so it's a very short half-life. However, the drug eventually crosses the blood-brain barrier, and we have shown it that it remains both in the muscles as well as the brain for 48 hours plus. So the effect sizes, you see it, 2 or 3 days after the IV infusion as well. And this is why the IV infusion is given on a weekly basis, not on a daily basis or twice weekly basis. Okay. Because as the slide says, that it creates a domino effect of physiological events. So it's a cascade of events that'll enhance the autophagy and lysosomal pathways to clear out these mutant protein aggregates, and hopefully, it'll help the symptomology of the patients. Next slide, please. What are the findings of this drug in the earlier patients that were treated? So one caveat we will always say as a researcher, that the earlier findings were all done in an open label, so no placebo was included. So that caveat should be, duly noted. And plus the small number of patients. These were not hundreds of patients. So, however, two studies, one was related to the previous sponsor where we acquired the drug from. So you can call it a related study. And another study was done in Asia, but that was completely unrelated to that. And what we found was both studies pointed to the similar findings. So in a way, kind of two different labs, independent findings. It's corroborate. And the findings were basically on the SARA scale, which is the scale for assessment and rating of ataxia. Most of the patients in the natural history, if they are not given any drug, they, over 12 months, both the European findings and the US findings have shown that these patients will worsen on SARA scale over 12 months, anywhere between 1.6 to 1.8 points. So they increase their baseline. And in our open label study done by our previous sponsor, they had 15 patients, 6-month study with continuation up to 1 year, where they basically showed they had taken 2 doses, 1/2 of the dose that current study is contemplating and 1/4 of a dose. So those were the 2 different doses they took. And overall, what they found was that the drug was generally safe and well tolerated with no reported serious drug-related adverse events. And more importantly, the patients were kept, when there was no meaningful change in the SARA total score over 6 months as well as over 12 months. And in fact, in one cohort, there was a slight decline, almost 0.5 decline of SARA scale from the baseline. So net, it pointed to improvement from the baseline, which was encouraging. But again, with the caveat of open label and small n. The other study that was done in Asia, they actually used a massive dose of oral trehalose. And I can tell you that amount of oral trehalose will not be allowed by the FDA or EMA. The Western regulatory agencies will not allow that, primarily because even in these 15 patients, 3 patients dropped out because of the massive GI events and side effects. So that was not advisable. However, in Asia, they did this study in SCA3 patients only for 6 months, and there was remarkable results overall out of 30. So 3 patients, I believe, were dropped out because of the GI events. And of those 12 patients overall, 8 of those 12 patients showed a remarkable 4 points drop on the SARA scale from the baseline. And so that was improvement rather than natural history worsening. And overall, the other 4 patients were stable to slight decline. And combined effect of those 12 patients were 1.8 points decline in SARA scale from the baseline over 6 months. So that was encouraging and pointing to very similar trend as our study. So that's the solid rationale that is the basis for us to be doing this late stage phase 2b/3 study currently, that we call it global. And Claudia will in a second give you the details of that study. And as I said, after that, we'll open it up to the questions. And to the extent we can discuss any of your comments or questions today, or you think of it later, do please come back to us, because we believe this is part of the community. It's a collaborative process. The more we hear your concerns and comments, we'll address that. And there's one minor point. I just want to make sure that although this study requires weekly infusion, we will provide an optionality to patients that on non-essentials day, that when they can't come into the clinics, they will have the optionality to get the home infusion. So on non-essential day, when there is no other scales are being measured, so we will allow that as well. With that, is that the next slide? Claudia, is that yours?
There we go. Thank you. The next one is mine. That is right. Thank you, Raj. Thank you for that great introduction and that background. Hi, everyone. I am Claudia Moore. I am the SLS05 SCA program lead for Seelos, and I will be going over a few other study details with you. Okay, so let's start with the study design. So in the study, we will be assessing the safety and the efficacy of SLS05 for the treatment of adults with SCA subtype 3, as Raj mentioned. So this is a double-blind randomized placebo-controlled trial, meaning that patients will be randomly assigned to receive the study drug, which is SLS05 Trehalose, or the placebo. And the patient, the physician, and even Seelos will not know if the patient is receiving the drug or the placebo throughout the study. I will, in a later slide, go over kind of how we plan to measure safety and effectiveness. But for now, let's stick to the study centers. So we're looking at including up to 30 study centers globally and up to 245 participants globally as well. And so the study period is a total of 56 weeks. So 2 weeks are allotted for screening. So all of that testing that we need to perform in order to determine if the patient is eligible for the study. And then 52 weeks of the treatment with, again, either SLS05 or the placebo, and then the 2-week safety follow-up. I do want to point out that once the 56-week period is complete, all patients will be afforded the opportunity to receive the study drug through an open label extension study. There it is. All right, so I kind of talked about how many patients. We're looking at 245. The study drug, again, is going to be SLS05, which will be administered through an intravenously. So it'll be either the SLS05 or the matching placebo, which is a sodium chloride injection. Raj mentioned this earlier. It is a weight-based dosing, meaning that the final volume that will be administered will really depend on the patient's weight. And the administration will occur over 60 to 90 minutes, and it will occur on a weekly basis. So once a week. Again, Raj mentioned something that I think is really important for patients, which is that we will be bringing on a vendor to support infusion administration in the patient's home. This way, the patients don't have to travel to the site on a weekly basis, because we understand that is a big burden. It's a big commitment. As far as randomization, so we have 2 major groups. We call Group A the higher dose group, and then Group B is the lower dose group. So the higher dose group, Group A, will include 200 participants. They will be randomized on a 1-to-1 ratio to either receive the 0.75 gram per kilogram dose of SLS05 or the matching placebo. Group B will include 45 participants, and they will be randomized on a 2-to-1 ratio to receive either the 0.5 gram per kilogram dose of SLS05 or the matching placebo. 2-to-1 randomization in here means really that for every 2 patients that get randomized to the SLS05, the lower dose, 1 will get randomized to the matching placebo. And then these cohorts will be enrolling in parallel, so at the same time throughout the entire study. All right, so let's talk about who's eligible by looking at key criteria for eligibility. So patients must be between the ages of 18 and 75 years. They must have a genetic confirmation of SCA3, their SARA score, so the scale that Raj mentioned earlier. That assessment, their total score on there at screening must be greater than or equal to 4. And then the gait component score must be greater than or equal to 1 at screening. And all patients need to be on stable doses of ConMeds for at least 30 days prior to screening. And then criteria that would exclude a patient would be, of course, anyone who doesn't have the genetic confirmation of SCA3. Anyone who has a score of 4 on any one of the 4 items that comprise the MSARA score, just because that is, the highest, most severe point for each category. Anyone who has a current diagnosis and or treatment of type 1 or 2 diabetes, or an HbA1c that is greater than or equal to 6.5% at screening. Again, Trehalose, it's a sugar. We don't know that it will, that something bad will happen, but we want to be proactive and preventative, and this is one way of doing that. And then, of course, any patient that has a medical condition or an abnormal finding during screening that could affect either the collection or how we interpret the data, or if it could be a potential safety risk to that patient, they will have to be excluded from the study. I did want to kind of highlight, so the MSARA is a 5-point scale that we developed in collaboration with the FDA, using the original SARA scale as a baseline. Most of you patients are going to be familiar with what the SARA scale is. Our MSARA includes the 5 components of gait, stance, sitting, and speech disturbance. The biggest difference is that, again, it's a 5-point scale. The SARA score alone, or the SARA scale alone, had a different kind of scoring system. Okay, so again, talking about how are we gonna measure if this is safe and effective, if Trehalose is safe and effective for treating specifically SCA3, because that's what we're looking at in this study. So to help us determine if it is effective for treating patients with SCA3, we will be relying on the MSARA assessment, the Clinical Global Impression of Severity Assessment, the Patient Global Impression of Severity Assessment, and the Frederick's Ataxia rating scale, the activities of daily living, all of which will be completed at baseline and then throughout the study and through week 52. So the MSARA and the CGIS will be completed by the physician or a qualified designee, while the PGIS and the FARS ADL will be completed by the patient. So we will really be looking for changes in these assessments and then comparing those scores at week 52 to the scores that are obtained at baseline, and that should give us an idea of kind of how the patients are doing and progressing. For the safety, we're going to be collecting adverse events. Every patient will be assessed for adverse events throughout the entire study. So we will be looking for incidences of treatment emergent adverse events, serious adverse events, including any clinically significant lab findings or electrocardiogram abnormalities. All right, so where are we located? So here is a map of the US. We have right now, we're working with 7 centers across the US, and we're actively looking for additional centers. We have 2 centers here that are currently recruiting patients. So UCLA out in Los Angeles, and then University of South Florida out in Tampa. I will move on to the OUS slide, but in the end, I will provide you with contact information if you are located in those regions, you want additional information about the study. Of course, we're also on clinicaltrials.gov, so you can find us publicly as well. But here we are in the OUS, we're in Brazil, we're in Portugal, in Spain, UK, Germany, China, Australia, and South Korea. So we have currently 17 centers outside of the US, and we are also actively looking for additional centers. All right, so here, as I promised, the contact information for the folks out in UCLA and in Tampa. And, if you want to recommend a center that is closer to you, or if you just have any general questions, about a center that may be opening near you, I do recommend that you reach out to us here at SLS05 team at seelostx.com. And with that,
Thank you, Claudia. This is excellent job. There were a couple of questions that I picked up the theme, and I wanted to address that from the chat room. So yes, there has been a prior data, animal model data of SCA3, but more importantly, there is a human data that I shared from SCA3, which is more relevant for us. Those were, again, small and open label, but very promising data. Wanted to make sure people know that. In terms of the right now, although there are only 2 sites open, the other sites that we listed, will be open soon. We'll be updating clinicaltrials.gov on a regular basis. So, please keep in touch with us. Please keep in touch through National Ataxia Foundation. We'll be sending out emails as more and more centers get open. And as Claudia suggested, to the extent, you have suggestions for other centers and those centers are cooperative in participation in such a study, we will be open to that as well. And in terms of one patient who has both SCA3 and SCA8, to the extent you have is very specific SCA3 identification, please present yourself to the sites, one of our sites, and we most likely will include you, but I don't want to say it a priori. I'll let the clinicians decide that, but we'll be open to that as well. The SCA6 patients, we are currently including them in an Australian study. So that's a kind of could be a geographical burden for you, but in the US, we'll have to wait, some time before we start that. So that's our response to the SCA6 patients. But Lauren, let's open it up to any verbal questions that come in as well.
Yeah, a couple more eligibility questions. There was one audience member who's enrolled in the troriluzole trial. Will that person be able to participate? Is there any age exclusion? And do you have how far progressed can your SCA be?
Got it. In terms of the trilozole trial, that should be fine. We should be, able to enroll because the concomitant meds, we are open to that as long as during this study, our study, there is a set, stable dose of that medication. In terms of the age eligibility, Claudia, what's the age eligibility?
So Raj, just wanted to clarify. So we do, there is the ability to be included. If you were on a prior clinical study, there is a washout period that would need to be done prior to inclusion. I believe it's 30 days. Again, this is all, you can see the inclusion exclusion criteria from this presentation, as well as on clinicaltrials.gov. There is the age cutoff of 75, and that is inclusive. So 76 would be considered excluded at this time for the study.
The question there, the one chat question, do we need to go to the sites weekly? So, and the answer is no, to the extent it's burdensome to you. We will provide optionality for home infusions. So you will be going to the sites for 3 months, 6 months, or 9 months measurement. And of course, the first 2-3 weeks when you initiate the study, but later on in between interim periods, we may provide the optionality to get you the drug through home infusion. Any other questions?
Was there a severity limit for inclusion, like a SARA score, a max SARA score for enrollment?
We have a lower limit. We don't have the upper limit. To the extent Dr. Biondi can see that question and respond to us, we will, otherwise we can, Lauren, can you keep that email handy and we will respond to them. I want to give them a comprehensive survey. So for example, certainly on the I think there is a one a sub scale where the scale is 0 to 4. If they are on 4, that'll be ineligible, and it's included in our eligibility criteria. But to the extent they are not, they are, let's say, at 3, they should be fine. But we can specifically, that's one off. Let's take it offline and we can address that.
Yeah, I could tell you that in the protocol itself, there isn't a maximum total score, but on the individual, as Raj mentioned, a 4 on any one individual component would exclude the patient.
Again, I see a couple of quick question. Who should be contacted regarding home infusion? It'll be part of the study, so you don't need to contact anybody else. You just need to participate and go through the process of the study, and the home infusion will be part of the study. And in terms of if you do not meet the age criteria of 76, is there any legal way to receive the drug? So currently, the only other place where we are doing this study is in Australia. But I don't know, Luke, whether we have a age criteria there as well. Luke, would you be able to enlighten us on that?
Yeah, currently the age criteria is the same for that as well. But again, as Raj said, if you are interested in the study, certainly reach out and we can, at least take your information. And if we are able to open the study to a broader age group, we'll be able to provide that information as well.
And that brings it to an important point. We want to make sure that people understand that after this 52 weeks plus minus study, it's a 1-year study, all the patients will be given open label extension. So any patients that were either on placebo or on the drug and they were showing a good effect, they'll be able to continue on that. And so with that regard, eventually in the future, but not now, we want to make sure if we ever do that, we may even do expanded access, the protocol down the road as we are contemplating for ALS. So that could be the way to include patients who do not meet the age criteria. But again, just keep in touch with us. But right now, currently, we are a small company. We are already doing like 3 or 4 other studies. And we are doing as much, and this is a global study. This is a big undertaking for us. But we thought based on the feedback that we heard from the patients, we thought it'll create a huge burden on Asian patients to be traveling to the US and migrating it for a year to participate in the study. So we thought it behooves us to just open it up worldwide as much as we can. And in terms, one question, Claudia, is coming up generally, patients who are interested in signing up for the study, who do they reach out to other than those 2 centers? Who do they reach out to?
Yeah, so they can, there's other centers listed on clinicaltrials.gov. I just posted the link right now in the chat. And then you can also reach out to the SLS05 team at seelostx.com. I can also put that in the chat.
Please
if you have
do.
any other inquiries.
Again. Yeah.
And I just want to also point out there's questions along with the home infusions. There's questions on patient reimbursement, and that is an option available at the clinical site. So if you speak to your investigator at one of the sites that are participating, they will work out reimbursement if you need to go to and from the clinical site, and we'll cover that.
Thanks, Luke. Another important question. I'm trying to put in as many questions we can address. So one question came out about genetic testing that the insurance companies has denied. So our thought will be work with the Please National Ataxia Foundation, and we will recommend sources to the extent. Anything else, Luke, you want to add about genetic testing if the patient?
No, Lauren and I want to know if you wanted to address that one.
Yeah, so earlier this year, we launched a free genetic testing program for individuals who are at risk for SCA1, 2, and 3. So what I mean by an at-risk individual, it means that you have at least one family member who has received a genetic diagnosis of either SCA1, 2, or 3. So if you are into that category and you're interested in getting free genetic counseling and testing, please email research@ataxia.org, and I'll put that into the chat. Yeah. So if you're interested in participating, but you don't have an SCA3 diagnosis, but have a family member who does, please feel free to use this free program.
Another question was asked. Thank you, Lauren, for that. Is this available to Mass General? Mass General is one of the sites that'll be opening soon. It's, again, there's a lot of paperwork and the process that goes through before the sites become open to enrolling patients. So stay tuned through us, through the clinicaltrials.gov, as well as feel free to ping them, the Mass General as well. But I think, we are in the process of opening them up as well. Another question, it sounds like Rahelos enhances autophagy, but has there been any indication of whether the increased autophagy degrades healthy proteins in addition to the mutant ATXN3? And the answer is, as far as I know, and we have done extensive, literature survey, as well as it does not attack the healthy proteins, because the healthy proteins will not have the excessive glutamine repeat sequences, and they will be folding into proper native helical structures. And as a result, they will never be toxic, and hence will never be a part of the autophagy lysosomal pathways. Good, thank you, Lauren, for putting up that email.
Hey, Raj, can anybody hear me? It's David.
Sure. Go ahead, David.
Yeah, I want to thank the team for filling in for me. I'm just getting over a case of pharyngitis and laryngitis. Regarding the SARA scoring, as Claudia mentioned, the modified SARA was at the request of the FDA. We worked together with the FDA, and we put together as Claudia mentioned, taking the 5 main items, the gait, stance, sitting, and speech. We have a 5-point scale, 0 to 5, 0 being normal, 0 to 4, 0 being normal, 4 being the worst. There is no upper score, but we're not allowing people eligibility who score 4 in any of those items. And these are the people that are most affected, right? So if somebody cannot walk, even with, strong assist of 2 people, that person would not be eligible. But right up to that point, the person is. The other thing that we have is that the person at least has to show some ataxia and gait. So they have to be 1 on that 5-point scale, and they have to have a total score of 4 out of the total 20 points that you can possibly get with the modified SARA. So it's pretty wide open for patients at all phases of their illness, based on the new scoring system.
Got it. There was another question Claudia was asked: when will the European sites will be available? What's the timeline for that, Luke?
That's going to be later this year. We're estimating sometime December, possibly January of next year. Mm-hmm.
Thank you for that. How good the trial results should be to go to the next state. So we are contemplating, if this study is comprehensive and positive, this could be enough, but ultimately for approval, but ultimately it's up to the regulators, both in the US, in Asia, and with Europe. So we are always at at their collaboration, and they look at the data, and then they determine. Nobody tells you up beforehand what will happen. And in terms of our mechanistic criteria, and the targeting, look, at the end of the day, there are 2 things we do well in research. One is we mechanistically try to focus on mechanism that should potentially give benefits to the patients. We do the preclinical studies, and then eventually the proof always comes as in the human studies. And our thinking is, in the human studies, that decline of SARA scale that the open label studies have shown in the past, that's a very strong rationale to pursue this because there is very few, if at all, any drug that have shown that. So that's the most encouraging thing. At the end of the day, mechanistic criteria is all hypotheticals, other than some, pathways that have shown to be beneficial, but the human data trumps everything.
There was a general question about overall timeline. Can you give an idea of how long it will be before you have results to report from this trial?
Generally, we expect 12 months for enrollment, and it's a 12-month study, so it'll be approximately 2.5 years before the data. And stay tuned. We will work on with the FDA, to see if they will allow us to do something else before that. But again, those are speculative right now. That should be the criteria. Another question came in: when will we open the studies in Brazil? We are planning to. What's the timeline, Luke, based on what we know today? And remember, anything we project today, that's our best guess today, and things happen, which is outside our control. Go ahead, Luke.
Yeah, so I believe we're looking into Brazil towards the end of this year, early next year as well. Claudia, there's also a lot of questions about how do we reach out to sites? What if a patient is not living near one of the centers there and they have a site recommendation? Whom do they reach out to, and how can we continue to give them this information?
Yes, so I included that email address in the chat, the SLS05 team at seelostx.com. That's if you have general inquiries, or if you have, again, the request to add a new site because there isn't one located near you. I see one recommendation for UCSF. I can tell you that I am in conversations with somebody there, so that's looking good for you. But yeah, email us there, or I will continue to update clinicaltrials.gov as well as we continue to activate sites. But please do reach out to the email if you have questions about when a site is going to come up. As Luke mentioned, right now we're estimating the rest of these US sites, the sites in Australia, the sites in Brazil, South Korea, and even a couple in EU to be up and active by the end of this year. So
great. Quick question, opening a site in India right now, we do not have a plan, but remember, the same drug is being studied in ALS, where we will have the data sometime middle of next year. And eventually, if we see a positive results in other indications, our focus will be to get the drug approved. And when we think about approval, we will not just think about the US, we're thinking globally, because these diseases are rare. And I think, we need to get this thing available. In terms of the other human data in other indications, other than what we are discussing today, Scott, it is interesting. If you look at it, the ALS is also, by the way, extra glutamine repeat in proteins. And we have mostly studied the data in the OPMD in the past. And OPMD is the muscular dystrophy, which manifests itself difficulty swallowing and eating, and where we have shown very positive data, but open label, where we help patients. Specifically, I remember there was a sibling, brother and sister. Both of them inherited the same gene mutation for OPMD from their parents, and the brother was in our study for almost 2 years, and he was able to eat and live his life according to him, what he told me, normally, where his sister, who was not able to participate in the study, did suffer from a very difficulty in swallowing and eating food, and was afraid of going onto the feeding tube. So we do have our drugs implications from other neurodegenerative diseases where it has shown an open label. So with a big caveat, help in improvement in walking, stopping them from falling, or improvement in swallowing. Whether that will translate into the SCA patients, the time will tell. That's why we are doing this study. But the earliest studies and other indications do provide a strong scientific rationale to do this study.
Okay.
A
Just wanted
quick
to-
question, do we believe that a weekly treatment will be needed for the rest of one's life? And the answer is, it may be needed, but we are working on other ways to get it to patients, the other formulations, which will make it much easier. But again, those are things that we work on, and it takes time to deliver it. So let's get the benefits captured first, then we can assure you, you will not have to go through IV infusion for the rest of the life if the drug works. Lauren, I interrupted you. Go ahead.
That was exactly what I was about to ask. So you covered it.
Thank you.
So there was a question about the pharmacodynamic biomarkers and leveraging that against using a placebo arm. And so we have taken that into consideration and discussed. The regulatory agencies are requiring a placebo-controlled study at this stage, which is why we do have these placebo arms, and we try to limit the number of patients receiving placebo arm within the study design. And again, for biomarkers, that is something we are considering as well further down and working along with the existing clinical trials for the natural history to leverage.
Got it. A quick question came in. They are looking for a closest site to St. Louis. Any answer to that? What's the site being contemplated? They'll be closest to St. Louis, Missouri.
Looks like Claudia might have to step away. So yeah, we are again evaluating these sites. There's not one right now, I believe, in Missouri. But again, if there is a site that may be able to work with travel and then home infusions, that might, you might not necessarily need a center right where you live.
Thank you. And just to, and the same person also wanted to know the home infusion reimbursement. If you're part of the study, the home infusion will be taken care of, so you don't have to worry about the reimbursement.
And you will also be reimbursed to go to the site for the visits that you do have to, the ones that have to occur in person at the site as well.
Correct.
So again, the focus is to, if you are the right person who qualifies for the study, to get you the therapy and hope for the best, rather than you need to worry about the reimbursements or the travel. We'll try to do as much as we can.
All right, well, we are past the hour now, so I just wanted to thank all of our speakers today and our really engaged audience. That was a great discussion, and note that this presentation will be uploaded to the NAF website on a new PrepRARE clinical trials page. Thank you so much.
We thank National Ataxia Foundation and Lauren and the team, and feel free to reach out to them, to us, for any other questions and comments that you can think of. And we are happy to work together with the community to get this study fully enrolled as quickly as possible. Thank you all.
Absolutely. Thank you, everyone.